Awesome. Thank you for an amazing talk, Liz. That was really inspiring. I've got lots of questions for later. But yeah, it's my pleasure now to introduce our next speaker, Bernice Waweru, who is currently a postgraduate researcher at the John Innes Center in the UK, where she's working on crop genetics and bioinformatics to improve under-studied crops and biodiversity. Before joining the John Innes Center, Bernice was working at the Kenya-based International Livestock and Research Institute as a bioinformatician, and she played a critical role in the sequencing and assembly of one of the first African-led genome projects for the underutilized crop, the lab bean. And I think she's going to tell us a bit about her role in that project. And yeah, I'm really excited to hear what she has to say. So with that, go ahead and take it away, Bernice.
All right, thank you very much. Hello, everyone from wherever you're joining. I hope you can hear me well. And thank you, Rose. Thank you, Will, for inviting me into this virtual conference. I'm happy to share some of the work that we have been doing. So changing gears a bit into some of the practical work that we've been doing to make sure or to enhance the work of African scientists in being involved in the work of taking advantage of the biodiversity that is within Africa and developing resources that scientists, colleagues and researchers within the African community, but also beyond, can be able to utilize for the African continent. So I'm just going to take you back a bit as I start my presentation today on where this all started in terms of crop improvement, taking you back a bit to the story of the father of the Green Revolution, that is Dr. Norman Borlaug, who began his research career a long time ago. And one of the key important things that he did was to introduce key technologies that were able to help a lot of countries to avert mass starvation after World War II through introduction of a lot of technologies. And he helped a lot of countries that were deficient in food to become self-sufficient in relatively short times, and thereby averting mass starvation. And one of his greatest legacies was in training that was able to make sure that whatever it is that was introduced was also sustainable and was also to be used continuously up to date. Unfortunately, it seems that Africa was left out of this revolution, and we are still hoping to catch up and still working. And the work that Ash and the other presenter have just presented is a testament to all the work that we need to do to improve this. So, sorry, my seems to just be acting up. Okay, sorry, that was not moving. All right, so Africa is growing at a very rapid rate, and it's estimated that 1 in every 4 people in the world will be living in Africa in less than 3 decades from now. Estimates on production say that we are going to hopefully catch up to the yield gaps, and that we will have caught up to the rapidly growing population. And for us to be able to achieve a balanced and sustainable food security, we must then be able to diversify our food resources to include underutilized crops that we can then help to increase the diversification of food production, to add new species to our diets that can result in better supply of nutrients, including amino acids, fibers, micronutrients, and the such. Out of all, about 370 roundabout known plant species, only 7,000 of those have been used pre-agriculture by human societies, and only about 250 of those are currently used as food species or currently cultivated. Of these, we only have about just 3 of them that provide about 50% of the global calorie production or calorie production and intake by the human population. And so for us to be able to achieve a more balanced and sustainable diet, we need to be able to diversify our food sources to crops that are native or indigenous to our continents and our planet. with this, I want to demonstrate that within sub-Saharan Africa, we have quite a diverse source of calories within Africa. We're able to produce most of it and currently importing a lot of wheat, of course, with the current pandemics and the current situations in European and Western countries. We are still very short on wheat and importing a lot of that currently. But on the other hand, traditional crops or underutilized crops, despite the fact that they are produced at subsistence level, we are still able to obtain quite a lot of calories from them, as well as protein from them, despite the fact that they are still grown at very much at a subsistence level. So if we are able to then take this up and scale it up to a level where they are grown to a larger scale, taking advantage of the traits from underutilized crops, then we can then be able to really diversify our diet and reduce the over-reliance on a few staple crops, and also be able to protect our environment by using or planting crops that are more adapted to our environment, and therefore also reducing the effect of our environment by using too much inputs like pesticides and too much fertilizers. And so low and middle income countries, including a lot of those in sub-Saharan Africa, we have a rich variety of these underutilized crops. And I want to focus my talk today on this underutilized, or sometimes called forgotten crops or underappreciated crops. Although most of them are very nutritious, most often crops are quite underutilized because they are underdeveloped. Most of them have long maturity periods, processing times, and some of them also have quite a high number of anti-nutritional factors. And I'm going to show this with an example called grass pea or Latharius sativus. It is highly nutritious, drought tolerant, but it produces a toxin, and when it is consumed for a very long time, it can even cause a disease that is called lytharism, and when eaten in high quantities can even cause death sometimes. But because of genomics, we are then able to generate technologies that can be able to avert such obstacles. And scientists have been able to generate a genome of grass pea, and they have now identified genes that are responsible for the toxins, and using technologies like gene editing to remove this toxin. So besides besides underutilized crop being very nutritious, they also have a lot of economic benefits. For example, when we look back to ancient times, a lot of these underutilized crops are the crops that were used during famine, during the war. For example, African yam bean was one of the crops that was used during the Biafra War in Nigeria in the 1967 to 1970 to provide a lot of the micronutrients, including protein and micronutrients, and also calories that were required for people to survive. And most of our crops are also used, or most of the underutilized crops are also useful for food and feed. So they are dual purpose crops for food and nutritious security. And of course, just as I've said, improving nutrition and as well building resilience in agricultural systems. So for example, other crops like fonio, bambara, amaranthus, they're also drought tolerant, and they are vital in drought prone areas, and very important to adverse climate conditions, and as well reducing that over reliance on just the very few staple crops that are grown. So when we use genomics to accelerate this development of these crops or forgotten crops, most of our countries within Africa and as well beyond, because there will also be benefit, we can then reduce the impact of importation, or even the impact that such importation costs have on our economies. So genomics as a whole has really changed the way that we do science, and particularly plant science. And a lot of platforms are now available. Most of them have their pros and cons, of course, but for low and middle income countries, some of them are more suitable than others. In this presentation, I highlight the Oxford Nanopore technology because it's one of those technologies that we really utilize within our work. And technologies like this, so one of these technologies like Oxford Nanopore, which is a third generation sequencing technology, opens up opportunities for low and middle income countries and the researchers and scientists to own and lead the research projects and research priorities within their countries. And so an example here is the Oxford Nanopore Min Ion being used in Kenya in one of the farms. So this is a farm for one of the ladies who grows cassava in their fields. And this was during a surveillance of cassava mosaic virus within Kenya, where we are able to detect the viruses and even identify which particular viruses there are. And this is very impactful because it then generates sustainable and impactful and tangible outcomes. And when we adopt such a mixed model that includes working with external partners to leverage these technologies, then this expertise will be grown within our local institutions to then take this to the other, to the next level in terms of working for priorities that are important for our African continents. So with that, I now want to focus on a bit of how we've been able to then leverage genomics and collaboration with external partners and collaboration with expertise from around the world to focus on underutilized crops that are of importance to our African continent, or at least the work that we have begun doing within the African continent. So getting away from helicopter science that was mentioned before, but then utilizing a north-south sort of collaboration where we are doing the work within Africa, all the capacity is being done within Africa, which will be really highlighted in the next talk. And then being able to focus on those projects and critical technologies that are important for us. So one of the crops I would want to talk about is lab-lab papyrus, which is commonly known as lab-lab. Dolichos bean, some of you might know it as Dolichos bean or hyacinth bean or black beans. So this is a dual purpose feed and food legume crop, which means that it can be used both for human consumption and also it's also an important crop for livestock. It's an important food for food, of course, for nutrition, for both feed, for both humans and for livestock. Its grains are quite rich in protein and micronutrients, and as well as that, the leaves can also be used as valuable sources of hay, as I've just mentioned, for livestock. And then they're also adaptable to a range of climates, soils, and they're also very good in terms of fixing biological nitrogen. So this work has actually already been published, and a lot of the work that I will mention here, I'm just going to go through it, but a lot of this can be found on a paper that we recently published. And as we can see, like what was just being mentioned, that hopefully we are shifting to a scenario where most of the first authors and most of the authors who are doing the work are from within the continent, as we can see within this paper. So most of those first authors are scientists and researchers from within Africa, and we were able then to collaborate with expertise to focus on priorities that are important for Africa. So the data for this genome lab-lab papyrus is available on NCBI under that bio project. So that's the genome, and then the genome and annotation is also available on EDAL. So anyone who's interested in looking at that can go to those links. So just to focus, or just to highlight a few of the tasks or roles that we were able to do within this project. Our goal was mainly to generate a long-read genome assembly that was not available. So initially there was a short-read assembly that was done by the African Orphan Crops Consortium, but it was not a complete or chromosome scale assembly. So what we were able to do was to then generate a chromosome scale assembly using long-read sequencing technology and to leverage what was already available or what had been done before. So what we were then able to to do was to use long-read assembly to generate a base or draft assembly. We then worked with phased genomics to orientate our scaffolds and to generate pseudomolecules of chromosome assembly quality. And the final assembly was about of a genome size of about 416 megabase pairs with 11 pseudomolecules or chromosomes with an N50 of about 38.1 MB. So we ran 2 independent pipelines to generate gene models. So one of those was the Fananushi pipeline that was run by the researchers back at ILRI. And then we also collaborated with some researchers from Germany to also run another independent gene prediction pipeline. And we combined those to generate about 24,972 high quality gene models. And from these gene models, we could be able to understand quite a bit of the lab architecture. So we generated a high quality genome. So just a bit of the statistics to our annotation and assembly, and looking at them based on the BASCO assessment results, we could see that we were able to achieve quite high completeness scores. So in terms of the assembly, we had about 98.5 completeness when we look at the genes that were present within our assembly from the embryophyta and Fabales orthologs or databases. And in terms of the annotation, which would be our genes, we also had quite high scores of about 97 and 94.9 for embryophyta and Fabales genomes. So when we also look at comparing the gene repeats, the high quality genome assembly and annotation, we were able to see some of the characteristics of the lab chromosomes as we have published. So we did an in-depth study of the repeats covering about 43% of the lab genome, and the LTR retrotransposons were the most abundant and covering about 28.1% of the genome. And we also identified tandem and unclassified repeats, which were found to concentrate and overlap at cluster points or peak points where they were breaking on the chromosome, indicating that some of the chromosomes at the centromeres is where we had a lot of the repeats. When we look at the pattern of the repeats and the genes, we were also able to identify that some of the chromosomes were metacentric, submetacentric, and some had subtelomerocentric chromosome structures. So furthermore, what we were able to do was also to look at gene family analysis of our lab genome, and we compared it with other closely related species. And what we could see, we found that we had about 14,047 genes that were unique to only the lab genome, as we could see in this Venn diagram right there in the middle. Those were the genes that we were able to identify specific to the lab genome. And from the lab genome, looking at those specific genes, the genes that were only expressed within the lab genome, from the gene family analysis, we could see a lot of the genes, what we could see that they were responsible for oxidation reduction, cellular component organization, catabolic processes, and furthermore, what we were also able to do was to then use our genome to look at some of the gene families that were expanded within our genome. So the high quality genome enabled us to dissect some of those traits, including the legume trypsin inhibitors gene clusters, which are protease inhibitors in that play a critical role in defense and in defense against pathogens and herbivorous insects. However, they are also known to be the anti-nutritional factors, and they reduce digestibility and availability of nutrients from the legumes to either humans or to livestock. And with the high quality genome that we were able to generate, we cataloged about 35 of these genes of the trypsin inhibitor gene family. which represented an enrichment of this gene cluster within the lab genome as compared to other legumes, including Medicago tranculata, Arabidopsis, Cajanus cajun, and Vigna angularis. So most of these genes we found to be located on chromosome 1, chromosome 4, chromosome 6, and two of the major clusters were present in chromosome 11, containing 21 of those genes. So with the high quality genome as well, we also worked with quite a number of scientists from the ILRI Gene Bank to look at the diversity of the lab accessions that we had there, and to perform a GWAS study to look at some of the morphological and yield related traits. So 125 lab accessions were genotype using DAT-seq markers, and they were also phenotype for quite a number of traits, and 18 significant markers or marker trait associations were identified across 8 chromosomes, explaining about 7 to 24% of the phenotypic variation. So this work showcases the high quality of the genome that was generated, and it goes to show that when we have such genomic resources, they are an important resource for studying and analyzing significant traits and underlying genes that can contribute to their improvement and breeding of underutilized crops, for example, lab like I've just shown here. And this research then goes into aiding adaptation, fostering wider cultivation, and maximizing the utilization of such underutilized crops. So a few of my next slides will just showcase some of the other genomes that we are currently working on and should be published sometime during the year. So one of those crops that the other underutilized crops that we're working on is Sphenostylus stenocarpa, which is Sphenostylus stenocarpa, which is, sorry, my timer just going off. So I'm just going to very quickly go through what we've done with this genome, which is also called the African yam bean. So this is a key nutritional security crop because it generates edible leaves, grains, and tubers that are highly rich in a lot of micronutrients, including proteins, magnesium, iron, among other things. And it also generates tubers which have significantly higher protein than more of the other common tuber crops, and as well as fixing also biological nitrogen, and it's adaptable to a range of soils and climates. So just as an example, this is from a recent paper that was generated that shows the estimated protein levels in African yam bean are way higher than that in cassava, sweet potato, or even Irish potato. So what we were able to do with this genome specifically was to utilize short read data and long read data to then come up with a genome that we were then able to also use with a lot of computer or bioinformatics tools to generate a base assembly. So because this is a work that was all done within ILRI and with African researchers, we were then able to test quite a lot of tools, and also that was also able to increase capacity. But we were also then able to work with Face Genomics to also orientate them into chromosome level pseudomolecules. And we generated a genome assembly that was about 649 megabase pairs with an N50 of about 69.5 MB. And with very high quality BASCO scores. So you can see there Embryophyta 98.5, for Fabalis about 98% for the genome. But also for the genes, we were also able to predict this from Fananotate, and we generated quite a number of genes which were also high quality. So we can see from the BASCO scores as well that the Embryophyta, from the Embryophyta genome database, we had about 91.1% completeness. One of the others that I'd like to highlight very shortly is this. It's a It's mostly used as a hedge in most African countries. It's called Dovialis cafra, and or commonly also called kayepo. So I don't know if most people know about it, but this is quite a crop that has recently also started gaining importance because its concentrate or a lot of secondary metabolites from its concentrate of the fruits are known to have anti-cancer, antimicrobial, and antifungal properties. So it's gaining quite a lot, and a few recent papers have recently been published, including one very recently in Nature. So with this, we were also able to generate a genome and some 27,000 protein coding genes. And furthermore, we also tried to look at the gene families from Dovialis cafra and also some of the secondary metabolites from it, just to see what's happening. And we could see that we have quite a number of those genes expanded from Dovialis when you did the gene family and expansion analysis. And just looking at the very top genes from those expanded genes, we could see that some of them are responsible for functions like osmoprotectants, which is important for drought resistance. And then we have jasmonate-induced stress response as well for climate adverse conditions. And also ajmalin and scopolamine are some of those key secondary or some of those processes that are involved in secondary metabolites. And so looking at some of those secondary metabolites that were produced or we were identified in the genome, we have most of them that are from saccharide clusters, but also from terpene and polyketide. And when we looked further, we could see that most of these are for flavanol. We could see that some of those are from terpene synthesis, and these are just very preliminary studies, but when we then go into looking further, we'll be able to generate this into a publication. But this is also one of those very key underutilized crop, or that has not been looked into, but which is a very, very important crop for Africa. So my take-home message would be today, despite the fact that we have a growing population, we are still able to work together, growing that capacity to then meet the challenge by looking at the wealth of crop genomics or crop diversity that we have, and then using that together with genomics and partnerships, learning together, and then putting those skills into being able to generate resources that are then going to be useful for Africa, but also for the world. Thank you very much, Rose. I know I've gone a bit overboard, but yes, thank you. I'd be happy to take questions at the end.
Great, thank you so much, Bernice. That was a really inspiring example of some African-driven projects on underutilized crops.